Hello and welcome to another lesson for Advanced Higher Biology. This one is in two main parts and is looking at hydrophobic and hydrophilic signalling. So what you need to be able to do is understand the different types of cell signalling. We're going to start looking at hydrophobic signalling before moving on to hydrophilic signalling. So as far as hydrophobic signalling goes, you need to know about the movement of hydrophobic signalling molecules. Describe what is particular about the receptor molecules for these signals. Give examples of hydrophobic signaling molecules, and explain how the hormone receptor complex is able to affect gene expression. There are two different types of signaling molecule. We have hydrophobic and hydrophilic. Hydrophobic molecules are lipid soluble, which means they're able to diffuse directly through the hydrophobic tails of the phospholipid bilayer. Other signaling molecules are hydrophilic, and so cannot pass through the bilayer in the same way. This means that they can affect their target cells in very different ways. A hydrophilic signal must bind to receptor proteins on the cell surface, and this sets off a signal cascade into the cell. Hydrophobic signals are able to diffuse across the membrane, along with perhaps diffusing across the nuclear membrane, and bind to an intracellular receptor. And it's these molecules that we're going to be looking at just now. In a hydrophobic signal, it's able to diffuse across the membrane, and when it reaches the cytoplasm of the cell, it's able to bind to an intracellular receptor. Hydrophobic signals are usually hormones, and when they bind to their intracellular receptor, the complex that results is referred to as the hormone receptor complex. The receptor molecule itself is a transcription factor, and that means it's then able to bind to DNA, and that can either stimulate or inhibit the initiation of transcription. The hormone receptor complex is able to bind to a specific DNA sequence, and that's called the hormone response element. Binding at those sites influences the rate of transcription and the corresponding binding of RNA polymerase. That allows this transcription factor to determine whether or not gene expression begins or doesn't. As these hormone receptor complexes are able to bind to these specific sections of DNA, it's possible for each hormone to be able to affect the expression of many different genes. The overall action of these steroid hormones, therefore, is that the steroid hormone passes across the plasma membrane, the hormone binds the receptor protein, which activates it. The hormone receptor complex binds to specific DNA sequences called hormone response elements, and binding at the hormone response element influences the rate of transcription. Hormones which act in this way are most commonly the steroid hormones, such as testosterone and estrogen. They all follow similar pathways and are able to act on many different genes. Estrogen, for example, is able to act on over 80 different genes. So you should be able to explain that hydrophobic signaling molecules are able to diffuse directly across the plasma membrane. The receptor molecules for these signals are transcription factors. That hydrophobic signaling molecules are commonly steroid hormones such as testosterone and estrogen, and that the hormone receptor complex affects gene expression by binding to specific areas of DNA and allowing transcription to go ahead or inhibiting that transcription of that particular gene. Next, we need to be able to look at hydrophilic signaling. So again, you need to be able to give examples of hydrophilic molecules and state whereabouts they bind. You need to be able to describe the 3 stages of hydrophilic signaling, describe the role of G proteins in relaying these signals, explain what's meant by a phosphorylation cascade, explain the action of insulin as an example of a hydrophilic signal, and explain types of diabetes in terms of insulin response and treatment. So quick recap, we've just mentioned hydrophobic signaling molecules and how they're able to diffuse across the plasma membrane. Now we're looking at hydrophilic signaling molecules which are unable to diffuse across the plasma membrane and must bind to a receptor molecule on the cell surface. This therefore sets off a signal cascade inside the cell. Examples of these types of signaling molecules would be our neurotransmitters and peptide hormones. So there are three main stages involved in hydrophilic signaling. First of all, we have a reception stage. This is where the signal molecule binds to the transmembrane receptor molecule. And as a result, the receptor molecule undergoes a conformational change. We then have transduction, and this is where the transmembrane receptor acts as a signal transducer by converting this extracellular binding event into intracellular signals. Our last stage is cellular responses. So these intracellular signals will invoke a response from the cell, and that may also exchange the gene expression. In the initial reception phase, the signal molecule, in this case we're calling it a hormone, is going to bind to the extracellular face of the transmembrane protein. This causes a conformational change in the transmembrane protein, and that activates it to stimulate a response from the cell. The transduction of this signal to the rest of the cell may involve either G proteins or cascades of phosphorylation by kinase enzymes. In transduction by G proteins, the G proteins are involved in relaying signals from activated receptors to target proteins. When we have the binding of the signal molecule to the receptor protein, that stimulates the subunit GDP to be displaced by GTP, activating the G protein, which is what's happened here. We've lost GDP, it's been replaced by GTP, and activated this G protein here. In turn, this G protein is going to stimulate the target protein, which is this one over here, and in this case is an enzyme. It may also be an ion channel. As a result, this activated target protein is then able to stimulate some kind of response from the cell. Transduction by phosphorylation, on the other hand, involves a cascade of reactions where one kinase enzyme activates the next in order to produce a cellular response. In this case, our transmembrane protein is a kinase protein, and that's inactive until it's bound by the ligand. The conformational change, which is caused by this binding, results in the kinase domains of the protein becoming activated. So there are areas of this protein which are able to become activated by this ligand binding. As a result, they are able to phosphorylate another molecule which has been present in the cytoplasm. This molecule is a protein kinase, which then goes on to phosphorylate a second protein kinase, and a third, and so on, until we end up stimulating some kind of intracellular response. This cascade effect allows one extracellular signaling molecule to activate more than one intracellular pathway. An example of this type of signaling is with insulin and the control of blood glucose. The presence of glucose in the bloodstream is detected by cells in the pancreas, prompting the release of insulin. As insulin is a peptide hormone, it's unable to diffuse across the membrane of its target cell, and so instead it must bind to the receptor site on its target transmembrane protein. These proteins are predominantly found in the cell membrane of fat and muscle cells. Binding of insulin causes a conformational change that triggers phosphorylation of the receptor. This then starts a phosphorylation cascade. This resulting cascade and signal will affect vesicles containing the GLUT4 transporter protein. This GLUT4 protein is responsible for being able to move glucose across plasma membrane. The vesicles containing this particular GLUT4 transporter protein is then stimulated to bind with the plasma membrane, and the GLUT4 proteins associate themselves with the plasma membrane of the cell. They are then able to transport glucose from the bloodstream into the cell via facilitated diffusion. Diabetes is a medical condition which results in an inability to control blood glucose level. Type 1 diabetes is caused by a failure to produce insulin, meaning the signal pathway is never started. This can be controlled by regular injections of insulin. Type 2 diabetes is generally associated with obesity and occurs when the cells no longer respond to insulin. Exercise is able to reduce this impact, as it also triggers the recruitment of the GLUT4 transport proteins by other metabolic pathways, and therefore improves the uptake of glucose to fat and muscle cells. So from that, you should be able to give examples of hydrophilic molecules and state where they bind. So you should know that these are peptide hormones and neurotransmitters, and that they bind to the extracellular face of the transmembrane receptor proteins. That the three stages of hydrophilic signaling are reception, transduction, and cellular response. That G proteins are able to become activated by the binding of the receptor molecules, and that they in turn activate a specific target protein. That in a phosphorylation cascade, the transmembrane protein is a kinase protein which is inactive until bound by the ligand. Conformational change results in its kinase domains becoming activated and phosphorylation of a molecule in the cytoplasm. That molecule therefore goes on to phosphorylate others and continues a cascade until an intracellular response is stimulated. That insulin is one example of a hydrophilic signal, as it binds to its receptor protein, setting off a cascade which results in GLUT4 glucose transporter proteins being associated with the plasma membrane of the cell, allowing it to move glucose into the cell and out of the bloodstream. That diabetes is a disorder which results in lack of control of blood sugar and is caused either in type 1 by insulin not being produced by the body, or in type 2 where the body no longer responds to insulin.